soil. And a lot of people don't quite understand that connection. So I'd like to go through an explanation of the biology in the soil and how it affects plant health, well, and the nutritional quality. So you understand why it's very important to have simple tools where we can be assessing the biology in the soil. So these microscopes cost all of about $250, and with the camera, another $50. We are working with ways to use your smartphone, and with a smartphone you basically can have exactly the same power that we have with this microscope. So you're not quite there yet. And one of the things we think of is that, you know, here in the first world, everybody's has the ability to get a smartphone, right? Well, maybe not. So how do we deal with people who don't have the money to afford a smartphone? Because a lot of smartphones are going to cost $500 as opposed to $200. So we've got some of those issues. But I want to go through an explanation of why biology is the determinant of soil health. Yes, I have the alphabet soup after my name, Whoopi. So yeah, a little bit of background on for me. I was the chief scientist at Rodale Institute. Some people aren't quite aware that I have left Rodale, so I just wanted to point that out. No longer there, back to Soil Food Web, based in Corvallis, Oregon, but we work all over the world. So I have laboratories spread across the world. It's fine to have these laboratories with big scientific equipment. We've got to move away from that. We have to put the power in the hands of each person. So how rapidly, how long does it take for you to take a soil sample, shake it up in a little bit of water, get a little eyedropper and put a drop of that solution onto your microscope slide and take a look at your biology. So we have quite a bit of training for those of you who might be interested. I have a brochure up here about our online courses. So yes, some on each side.
Excellent, thanks.
Because you can go online and get the whole lecture series about the organisms in the soil, compost, because that of course is how we're going to put this proper biology back into the soil. Compost teas, compost extracts. If it's hard for you to apply a solid, we're going to turn it into a liquid. And then microscope. So you can learn all of these things online. So pretty briefly, I want to, 15 minutes, oh geez.
You can do it.
I'm going to talk really fast. What is soil? And most people aren't really quite certain what the difference between soil and dirt actually is. So you've got to, when you say that word dirt, it's the 4-letter D word in soil science, so you must say it with that little sneer on the lip. You know, that's very important. Dirt. As opposed to soil. The father of soil science, Hansiani, defined soil as having these components. The mineral components, the sand, the silt, clay. Where does the sand, silt, and clay come from? Well, it's the breakdown of your rocks and gravel and pebbles, your parent material as it's broken down into smaller and smaller chunks. And back before there was life on this planet, the only thing that broke down parent material and turned it into the smaller and smaller bits was freezing, thawing, wetting, drying cycles. But as soon as life started to occur on this planet, the most important thing in breaking down your rocks into all the smaller components are actually the bacteria and the fungi. Quite a bit of work going on at the University of Helsinki in Finland showing that that is actually the case. It is life, the organisms, that do that job of making sand, silt, and clay. Sand, something you can see with your eyeballs. Silt, which is the size of a red blood cell. And clay, which are the same size as bacteria. Luckily, clays are tattered and frayed along the edges, so you can reasonably well differentiate them from bacteria. So the mineral component, organic matter, and organisms. Hansiani didn't overtly say that it has to be aerobic, but Hansiani was very clear about the fact that you can't have waterlogged soil. Something happens when your soils go anaerobic. That's not soil anymore. It turns into putrefied materials and you're going to kill your plants. So it has to remain aerobic. Soil is, by definition, an aerobic material. So when we start trying to understand each of these components, I want to go through them pretty rapidly. When we're looking at the sand, the silt, and clay, we're talking about all the mineral nutrients present in your soil. And in every single introductory soil science textbook, every single agronomy textbook, there are tables like this that show all the mineral nutrients, and I've only got the top 17 here, so yes, we've got the rest of the 100 and whatever it is on the periodic table. But what I want to point out is that there is no soil on this planet that lacks the nutrients to grow plants. There is no soil that lacks the nutrients to grow plants. Why are we putting inorganic fertilizers out? Why do we think we have to put those toxic materials? Every single inorganic fertilizer is a salt. It's going to kill the organisms in your soil when you add those fertilizers at high concentrations. All the nutrients that you need. Do we need nitrate fertilizers in our soil? Look at the nitrogen content in your sands, your silts, your clays, your rocks, your pebbles. That is more than enough nitrogen per teaspoon of your soil to grow all the crops in your whole entire acre. Why are we putting on inorganic fertilizers? Because we killed the biology. Through the applications of these inorganic fertilizers, you kill the biology that will convert these nutrients from the total mineral form into the plant available form. How does Mother Nature do this job of taking the nutrients which are present in your soil? Nothing's lacking. How does nature take these nutrients and convert them into that plant available form? And it's all about life. So we want to make sure that we get these organisms back into our soils. So you don't have to worry about the fact that your soil is too poor to grow corn, too poor to grow bushes or shrubs or any crop that you want to talk about. The nutrients are present. I do want to point out the next column over is where we're looking at the range, the variability as we go to soils all over the world. Every single major soil on the planet has been summarized. There is some range of variability, but even at, for example, with nitrogen, 200 parts per million nitrogen is still plenty to grow most crops. The problem is, as we've gone through industrial agriculture, we kill those organisms by tillage, by the application of toxic inorganic fertilizers, and then of course, pesticides. So need to get that biology back into the soil. Why do we see plants grow better when we put inorganic soluble nutrients back into the soil? Because we already killed the life. There is nothing to do that conversion, and so you are forced to put in the soluble nutrients. So which is better? Constantly having to add inorganic fertilizers. 80% of every inorganic fertilizer we add into the soil, that nutrient is going to leach out into your groundwaters, your drinking water, your surface waters. 80% of every inorganic fertilizer that's put out within 1 year has leached into groundwater, drinking water, surface waters, etc. How much are we harming ourselves? Okay, I think you can figure that one out. Get the life back into the soil instead. When we take a look at that total nutrient, that's the blue box. It is the bacteria, fungi growing in that soil, immobilized and holding nutrients in their bodies, being eaten by the protozoa, the nematodes, the microarthropods, the earthworms. And that's what releases plant available nutrients. So the soluble pool, if any of you know Albrecht's work where we're looking at how do you flocculate clays, and I'd love to go into that topic with you, but if you don't know what I'm talking about, the online course would be a really good idea. So how do you get your soils to build structure? You've got to have these organisms which allow that to happen. And then the next step, releasing nutrients in a soluble form, and look at what percent of the total pool, the soluble, the inorganic soluble pool is. When you send a sample into a chemistry laboratory, into a soil chemistry lab, which one of these pools did they tell you about? How can they possibly convince you that you have to buy inorganic fertilizers if they tell you about the total pool? No one would ever do it. But because they only tell you about this pool, yes, that is the pool that your plant takes up. But they're not going to tell you about that process by which you're going to liberate the nutrients that are present in your soil and turn it into plant available forms. It is the organisms that do that work for you. So the next component within our soil is the organic matter, and just to make sure that you understand what organic matter is, I enjoy that we have a competing seminar where one of the things they were going to talk about is what is organic matter in soil. Here we go. Isn't that difficult? It's dead plant material. Plants also produce exudates from their root systems. When you're looking at a tree, 85% of the photosynthate produced above ground by a plant, by a tree, moves down into the root system, stays in the root system. 85% of the energy produced by a plant goes down into the root system. 50% of that energy is going to be released as exudates to feed bacteria and fungi. So all these wonderful cakes and cookies is what we talk about, because that's really basically what we're talking about. Mostly sugar, a little bit of protein, a little carbohydrate. So cakes and cookies, and hundreds of thousands of different kinds of cakes and cookies coming out of that root system to grow hundreds of thousands of different species of bacteria and fungi. The plant is essentially telling the bacteria and fungi growing around its root system, hey, I need some iron, go out and find it for me. Now I need some calcium, go out and find it for me. So the bacteria and fungi, using those foods the plant is feeding them, makes enzymes that break down the structure of the sands, the silts, the clays, the rocks, the pebbles, and oh yeah, organic matter too. So some of that organic material, plant exudates, is specifically meant to grow the specific bacteria and fungi to do the work for the plant. Go collect those nutrients. But of course, the bacteria and the fungi get those nutrients. They're right there, they're the ones that made the enzymes, so they suck up those nutrients and hold them in their body. It's the pantry for your plant, retaining and holding nutrients. If we've got good amounts of bacteria and fungi in our soil, we will never have any leaching out of that soil. That's how you make clean water, is to make sure that all of those soluble inorganic nutrients are sucked up and held by the bacteria and fungi being fed by your plant. So here's all our nutrients and our bacteria and fungi, and now we have to have the predators in the system. The protozoa, the nematodes, the microarthropods, the earthworms, they eat the bacteria and fungi right around that root system and release those nutrients right there at the surface of the root. And in all of the balance, not just nitrogen. When you put on a nitrate fertilizer, you're only putting on one type of nutrient. Is your plant going to grow on one nutrient alone? No way, Jose. So we have to have the whole balance. What does your plant need this second? Well, it's something else now, and it's something else now. And your plant needs something else now. Every second of every day, that plant has to be given the proper balance of all the nutrients. Your plant will not be stressed. It will not be subject to disease. It will not be subject to pests. When we get the balance of this biology in the soil right, weeds don't grow. Because weeds require a very early stage of succession, and we want to move that biology in our soil beyond that early stage of succession so we can grow the plant we want to grow without having weed problems. So say goodbye to pesticides, say goodbye to inorganic fertilizers. Not necessary, not needed. So bacteria and fungi doing decomposition are modifying these nutrients. Waste released by one set of organisms is food for a different set of organisms. So complexity within our soils. When you look at all the different kinds of organic matter present in our soils, well, of all these different kinds of foods in our soils, now the really simple structures, these are going to be foods for bacteria. But when we get into really complex structures, it is the fungi that make these structures, and it's the fungi that use these structures. So what's that ratio of bacteria and fungi? And that's what helps select for different plant species, which species are going to grow, because we're setting the stage for the growth of these different plants. And I wish I had time to go over all of that, but
You have 5 minutes.
I've got 5 more minutes. I think I can manage that. Organisms. So how do you know that you have your organisms in your soil in the proper balances? Do you have enough bacteria? Do you have enough fungi? Do you have enough protozoa, nematodes, microarthropods, earthworms? How do you know? Well, earthworms, great. You dig up a square foot of your soil and you sieve the earthworms out and you count them. That's pretty easy. But how do you know how many bacteria? It's going to take a microscope for us to take a look at. So when we try to understand that nutrient cycling system, soil food web, we have to be able to use a microscope and assess these organisms present in the soil. So just briefly, I've gone over the various things that all of these organisms in your soil do for you. Suppress diseases, we don't need pesticides. Retain nutrients, you don't have to be adding inorganic fertilizers, making those nutrients available to your plant at the rate the plant requires, and of course, who's really in control of the rate at which those nutrients become available to your plant? It's the plant. Don't you be bothering about, do you have enough nutrients in your soil? Your plant's going to take care of it. Our job is just to put the diversity of the organisms back into that soil. And so that's where this microscope gets to be very important. Do you have enough? If you don't, how are you going to add those organisms back into the soil? You're going to make compost. Actual real compost, not putrefied organic matter. So we can take a look at your compost and make sure you're going to put back in the organisms you require. And then once you have added that back into your soil, take a look at the soil after a week. Have those organisms survived? Are they there? Are they present? If not, then you put some more compost in. Until you get the organisms up to the proper balances, your plant's probably not going to be healthy. Building soil structure. It is the bacteria and fungi that make the aggregates. It's the protozoa and nematodes that move those aggregates around and build pore spaces. We want water to infiltrate into the soil, and if we rebuild structure, we decrease water use by 70% in our soils. In California, we're doing that over and over and over again. Around the world, we've reduced water use in South Africa, in Australia, in Indonesia, and all over the world. We have to get root systems going deep into our soils. Most people think roots of plants only go a couple inches, a couple of feet. Absolutely not true. If you've got a compaction layer, that's going to kill your roots. The processes that are going on in anaerobic conditions will kill your root systems. And so you have to break up that compaction. You have to rebuild that structure. That requires oxygen. You have to rebuild structure to let oxygen move as deep as it can into the soil. How far down into the soil surface do we find aerobic soils? A lot of people, because they've been fed misinformation, will say root systems only go down a couple inches, a couple feet. Well, in fact, we're working with some of the old growth forests in the Pacific Northwest, and the root systems of those trees go down about 300 feet. How far down do we find oxygen into the surface of the planet? 16 miles. Soil is not anaerobic as you go down deeper. If you've got good structure, if you've got the organisms working and functioning. Do we have active living growing organisms at 16 miles? How about in molten rock? Are there active living growing organisms in molten rock? Yes, there is. There's research being done at the Macaulay Institute at the University of Aberdeen, showing that there are bacteria growing in lava, in molten rock. So how far down do you actually find life on this planet? Probably all the way to the core. So just because we've never looked, just because we've not had the methodologies to look at these things does not mean it doesn't exist. So technology, ways to look at this, not requiring you sending samples into a laboratory, do it yourself. So just a quick look at some of the different critters. Can you determine which are the beneficial organisms in this picture and which are the bad guys in this picture? Can you find the root? And can you tell if this root is healthy? And if you understand the biology, if you understand what each of these different organisms are doing in this picture, you can tell immediately you're in deep trouble. You're going to lose your plants in the next week or two. Your whole system's going down. Disease is going to wipe you out because your soil is going anaerobic. You've got disease-causing organisms attacking this root, and you might want to learn about that. So when you're looking at that root, see the divot out of the side? Your root system is being attacked by a disease-causing fungus. This ciliate, see the little cilia on his body? That's how you identify ciliates. It's an anaerobic organism. Your plant's gonna be dying in a not too far distant future. So if you're looking at your soil using this technique, you can tell when you better get to work right now to bring your plants back into a condition of health. Fungi in the soil. Can you identify the 4 different species of fungi in the soil? And is it important that we have that much diversity? Absolutely. Because every time we're at a different temperature, different moisture, different calcium concentration, iron, zinc, boron, all of those nutrients, every time things change a little bit in the soil, a different species of bacteria and fungi, different species of bacteria and fungi will wake up, everybody else goes back to sleep. In a healthy soil, we ought to have 75,000 species of bacteria and 25,000 species of fungi, about 1,000 species of protozoa, and about 100 species of nematodes. They're not all awake and functioning at every second, but in order to protect our root system against diseases and pests, we have to have a portion of that community actually active and functioning. So can you pick out the bad guy fungi in here? Can you pick out the good guy fungi? So good guy fungi, a little out of focus, so yes, you have to be able to focus with your microscope. We have another fungal hypha right here. We have another fungal hypha right here. Clear, colorless, not as good as something that's nice and dark in color, but diameter is also important. The disease-causing fungus in this picture is right here. Very narrow diameter, clear, colorless, that's Pythium. So how do you tell immediately whether you've got the good guys or the bad guys? And using the microscope is the way you're going to be able to do that in 5 minutes. So helping people learn this technology. Can you pick out the fungus in this picture? Hopefully by this time you can. Yep, and lots of bacteria in there. We've got all kinds of different sizes and shapes of bacteria, so understanding all of this. Looking at mycorrhizal colonization of your root system. You take your roots, you wash your roots off, you put it on your microscope slide, you illuminate it with an ultraviolet lamp, and the arbuscules, the active exchange between the fungus and the plant, autofluoresces. So we have approximately 44% colonization of this root system with mycorrhizal fungi. We know our plant's going to be doing well. So when we lose that colonization, we know we're in trouble. Ectomycorrhizal fungi, understanding these organisms and teaching people who they are. How do you identify them? Can you find a flagellate? Is a flagellate a good guy or a bad guy? Beneficial organism. It eats bacteria and releases nutrients in a plant available form, and the whole balance of all the nutrients your plant's going to require. So identifying is what we help you learn how to do. Is this a good guy nematode or a bad guy nematode? How do you tell? Look at its mouth. This guy is a filter feeder. It just filters the bacteria out of that soil solution. How about this guy? This is a fungal feeder. Has a spear. It's going to stab that fungus and suck out the internal contents. Isn't soil a nice place?
Wow.
That's scary, isn't it?
You could make like a movie out of it.
Yeah, I want to make a cartoon. I would
Totally.
so love to get some funding to. Yeah, I've got the stories half written.
It'd be awesome.
Remember, it's a bug's life. We could do a couple of those. So all the wonderful things going on.
Who
And
wants
so,
to fund that?
yeah, no, please. I'd love to do that.
They all have different personalities.
We got to have Freddy fungus and Betty bacteria, and there's got to be a love story involved. And the alien, the invaders come, the anaerobic organisms. Oh man, we could have so much fun. And then kids would understand. So I need an artist, somebody. So okay, predatory nematode. Ooh, most people assume a predatory nematode's going to be bad because it's eating things. No, these are beneficial organisms because their absolute favorite food are root-feeding nematodes. The things that destroy about $5 billion worth of plant production on an annual basis in the United States alone. Root-feeding nematodes. Can you pick out the distinguishing characteristic of root-feeding nematodes? So when you look at the spear in its mouth, the big spear, see the big knobs? Brood-feeding nematodes are the only thing that have knobs like this. So when we teach you these characteristics, you very rapidly can start doing these kinds of identifications yourself. It takes about a day for us to teach you how to use the microscope and how to identify these different organisms. You know, we do not need 3 decimal place per accuracy. What you need to do is say, do I have enough fungi? Do I have enough protozoa, enough nematodes of a good variety? And those are the sorts of things that we want to teach you how to do. And we've got online courses to do that. Organisms do all the work in soil. And that's something that no one was taught back in grade school. Even today, most people are never taught that it's the life that's doing the work. It's the life in the soil that's making certain that your soil is healthy, that do all these positive things for you. So during the demonstration phase, I want to actually go through and show you how easy this microscope is to use.
Perfect. Thank you.
15 minutes.